Hey team, welcome back. I'm glad you're here. So we've talked about mitosis. We've talked about what happens when the regulation of that cell cycle doesn't function properly. Now we're going to talk about a different type of cell division called meiosis. Meiosis. And this is the type of cell division that creates gametes. So sperm and egg cells in organisms that are reproducing sexually. All right, so meiosis is important because it creates 4 unique daughter cells. Those 4 daughter cells are all going to be genetically unique from each other, and those 4 daughter cells are going to be haploid, right? So for most of our cells in our body that we call somatic cells, so soma means body. So for most of the cells in our body, with the exception of our gametes, our cells are going to be diploid, meaning that they're going to have 2 copies of each chromosome. Meiosis, this type of cell division, meiosis creates haploid cells that are only going to have 1 copy of each chromosome. And that's significant because once that one haploid cell comes along and finds another haploid cell, they'll combine in the process of fertilization and they'll create a new diploid organism. So meiosis is the process that generates these haploid cells that are used for sexual reproduction. And sexual reproduction is a big deal because it helps to introduce genetic diversity into a population. So that way all individuals are not the same. Individuals are unique genetically from each other, which is going to be important when we talk about natural selection, because that genetic diversity, that difference in those genes, allows populations to evolve and adapt to their surroundings. So big picture here, meiosis creates haploid gametes. We're going to go from 1 cell to 4 haploid cells that are all genetically unique from each other. They're all genetically different. Okay. So let's take a quick look at what DNA looks like in a somatic cell. So this is what we call a karyotype. So this is an image of DNA, and it's organized into the chromosome number. So this is an image of a human female. And notice that for chromosome 1, there's 2 chromosomes. Same thing for 2, 3, 4, 5, so on and so forth. Every chromosome, there are 2 chromosomes. They come in pairs. So we have 2 pairs of chromosome 1, 2 chromosomes 2, so on and so forth. Okay. So this is a diploid cell. Diploid meaning having 2 sets of each chromosome. Okay. One of these chromosomes in this individual came from the mother's egg. The other chromosome came from the father's sperm. All right. So in each of us, 1/2 of our DNA comes from our mom. The other half comes from our dad. Right. And so what meiosis is going to do is meiosis is going to separate these homologous chromosomes. So these 2 pairs right here, these 2 chromosome 1s, we call these homologous chromosomes, because they contain the same type of information, just maybe different forms of that information, right? Maybe this one right here says you're going to have, your second toe is going to be longer than your first toe, than your big toe. And this one says right here, your second toe is going to be shorter than your big toe, all right? So the same trait, toe length, just different forms of that trait, right? Or we can call these alleles as well, which we'll talk about alleles more in depth next week. Meiosis is going to separate these homologous chromosomes to create a haploid gamete. Okay? And gamete is just a technical term for sex cell, a sperm or an egg. So if you haven't watched the mitosis video yet, or we haven't reviewed mitosis, go ahead and stop here. Go watch that video, because we're going to be using a lot of those same terms and those ideas again here in this subject. All right, so meiosis. What's neat about meiosis is basically the same things are happening in meiosis as what's happening in mitosis. We're just going to have 2 rounds of division, right? There's going to be some slight differences, especially when we talk about meiosis 1. There's going to be some things that are going to be significantly different in meiosis 1 compared to mitosis. But we're going to have the same phase, prophase, metaphase, anaphase, telophase, cytokinesis, and then we're going to go through another round again. Prophase 2, metaphase 2, anaphase 2, telophase 2, cytokinesis. So if there is a number, here's a big hint. If there is a number associated with a phase, let's say I ask you, what is happening in anaphase 2? If there's a number associated with that phase name, that means it's referencing a meiosis phase. We only number the phases in meiosis. We do not number mitosis phases. So meiosis 1, prophase 1, metaphase 1, anaphase 1, telophase 1. Meiosis 2, we're going to repeat those again. Prophase 2, metaphase 2, anaphase 2, and telophase 2. All right, so now we're going to start into our overview of what's happening in meiosis 1 and meiosis 2. So we're going to start with a diploid cell that has already gone through the S phase. It's already gone through the G phase. So we're starting with a cell that has already duplicated its chromosomes. So here we go. So here we're talking about meiosis 1. Here's the DNA. So the DNA, it's in that X shape. So it means it's already gone through S phase. So that means that these sister chromatids are attached at these centromeres. Right? So DNA is already replicated before we begin meiosis. So in meiosis 1, the first round of prophase, metaphase, anaphase, and telophase, or PMAT, in this first round, the major differences occur, especially during prophase and metaphase. So in mitosis, when we're just making identical copies, all of the chromosomes line up in one straight line. So we'd have 46 total chromosomes lined up in one straight line. In meiosis, the chromosomes are going to pair up in their homologous pairs, right? So remember, homologous pairs are these two right here, 2 chromosomes number 1. These would be homologous pairs. These would be homologous pairs for chromosome number 8, right? So the chromosomes in meiosis, in prophase, the homologous pairs start to line up next to each other. So chromosome 1 you got from dad and chromosome 1 you got from mom, they line up next to each other. And then in metaphase 1, they line up in what we call a tetrad formation. All right, but why are they doing this? And why are they lining up in homologous pairs like this? Well, that's because something unique happens in meiosis 1, especially in prophase 1. This only happens in prophase 1, called crossing over. So crossing over, so when these tetrads form, so here we have homologous chromosomes, when they pair up into these tetrads forms, what happens is parts of those sister chromatids from the other pair, from the other homologous pair, part of those sister chromatids wrap around each other, and then the DNA swaps places. So here we go. So here's our homologous pair right here. They're going to cross over. They create this connection right here, which we call a synapsis, and then they swap places. So see, this chromosome prior to this process was all blue. Now look at this chromatid. It's blue, but now it has this little bit of red right here. Right? And then same thing right here. This chromosome, this chromatid that was all red, it's got a little bit of blue right here. Right? So crossing over, swapping DNA from these sister chromatids, what it's doing is it's mixing up the genetic information. So it's mixing up genetic information and resulting in combinations of genes that have never been seen before until this point in time. But why does this matter? Why does this happen? This happens because the whole point of meiosis is to provide cells for sexual reproduction. And what's the point of sexual reproduction? The point of sexual reproduction is to increase the genetic diversity of a population, so that way the children and offspring are not identical to each other. Because diversity, genetic diversity, is one of the main drivers for natural selection. And this process of crossing over helps to generate genetic diversity, because we're getting new combinations of genes that are being swapped around. Okay? Crossing over helps to generate genetic diversity. So now this combination of genes right here, this has never been seen before. So another unique thing that's going to happen in metaphase 1 of meiosis 1, how those tetrads line up, so we call those homologous chromosomes that pair up during meiosis 1, we call those tetrads because there's 4 sister chromatids together. Tetra means 4. So how those tetrads line up at that metaphase plate, that middle region in the cell, where all the spindle fibers are pushing those tetrads together, the orientation of how those chromosomes line up is going to vary, and it's completely random. So like, for example, right here in this cell, we have all of the red chromosomes on one side, all the blue chromosomes on the other. That results in the daughter cells, 2 of them having all blue and 2 of them having all red chromosomes. But what if we swapped it? What if we did red, blue, right? That results in 4 different cells. These ones have one large blue chromosome and one small red chromosome. These ones have one red large chromosome and one small blue chromosome, right? So take that orientation of how those chromosomes line up for all of our 23 pairs. That's completely random. That's going to result in completely unique gametes that are formed. And this is why siblings from the same parents can look completely different from each other. Crossing over, swapping of genes, as well as this random arrangement of chromosomes of these tetrads during metaphase 1 results in completely unique gametes being produced. So same phases, we're going to go through prophase, metaphase, anaphase, and then telophase. But the difference in anaphase and meiosis 1 is that the homologous chromosomes are separated. In anaphase of meiosis 1, we separate the homologous chromosomes. So anaphase 1 separates the homologous chromosomes. Telophase and cytokinesis separate those two cells. So at the end of meiosis 1, we have two cells that are each haploid, but we still have these sister chromatids. This is the cell what the cell would look like at the end of meiosis 1. So each chromosome still has two sister chromatids. So meiosis 2 is going to separate those sister chromatids. So in meiosis 2, we have the same thing, prophase 2, chromosomes condense. Metaphase 2, chromosomes line up at the metaphase plate, that middle region, but now they're going to line up all in one line. All the chromosomes line up in one line in metaphase 2. And anaphase 2 is going to separate the sister chromatids. Anaphase 2 separates the sister chromatids. And then telophase and cytokinesis results in four total haploid gametes, haploid cells, that are all genetically unique. They're all genetically unique. So they're haploid, so they have one of each chromosome. So in humans, our diploid number is we have 46 chromosomes. Our haploid gametes have 23 chromosomes. So we get two 23 chromosome cells recombined together, and we're back to our diploid number. All right, so that's meiosis. We'll talk about what happens when this process doesn't function properly in our next video. All right, good job.